Hello, thank you for tuning in to our presentation. As you've probably seen on the conference webpage, this presentation is about if pollen can be useful in archaeological studies. So during this presentation, we will show you and explain what is pollen, why are they useful for your work, how to study pollen, and at the end give you an example of a case study from a real archaeological site. My name is Friðgeir Grímsson. I'm a senior scientist at the Department of Botany and Biodiversity Research at the University of Vienna, Austria. We are housed at Renweg 14, that's very close to the Botanical Garden. As you can see, I'm standing there right now. At the department, I am responsible for the electron microscopy labs, teaching courses related to electron microscopy and palynology, and leading the palynology research team. Along with my team, we study fossil, subfossil, and recent pollen from all geographical regions and ages. But important is to us is that the sample has an interesting background story. In botany, we refer to pollen grains as the male gametophytes of seed plants that produce sperm cells. Pollen are produced by both gymnosperms and angiosperms as part of their sexual reproduction cycles. In gymnosperms, pollen is produced in so-called microsporangiate cones. Most gymnosperms are wind-pollinated. As an example, we see here a bee-saccate pinnaceid pollen. In angiosperms, pollen is produced in anthers that are part of flowers. Most angiosperms are either wind-pollinated, animal-pollinated, or both. As an example, we see here a triaperturate tilia pollen. What is important to know that pollen grains have a wall which serves to protect the gametophyte during transport. The pollen wall is divided into two main layers. An inner layer called the intine that is composed of cellulose and pectin, and an outer layer called the exine composed of sporopollenin. Now, the inner layer, the intine, is easily dissolved and disintegrates, but the exine composed of sporopollenin is extremely resistant even to various different dissolvents. It is thanks to this outer layer, the exine, composed of sporopollenin, that we are able to investigate dispersed pollen belonging to archaeological samples, forensic samples, and paleobotanical samples. Due to the resilience of the pollen wall, pollen grains can be preserved for millions of years. An example here would be this fossil Gunnera pollen that's about 82 million years compared to a recent pollen grain from Gunnera in our botanical garden. There is a good reason why pollen should be a potential target in your research. And that is, pollen is found more or less everywhere. So plants, they actually need pollen in their reproductive cycle. So we have pollen are being transported from the male organs of the plants to the female organs. Now, this is very often done by wind. And there are several plant species, especially trees. They produce a lot of pollen. And in one large tree that is maybe 30 meters high can have hundreds or thousands of flowers. And then you can have tens, hundreds, or even thousands of trees. And then you multiply this with the number of pollen produced by each flower. You can see that during springtime or early summer, you can have millions and millions and millions of pollen in the air. Now this pollen, even though we don't see it, is all around us. It gets in our clothes, it gets on our shoes, we get it in our hair, we get it into our cars, we get it into our apartments where we live, it's in our kitchen, it's in our bedroom, it's in our beds, it's in our bathroom, it's everywhere. So literally what I'm saying is that we eat, drink and breathe pollen all the time. The reason why pollen is useful for various different scientific disciplines is that the morphology and ultrastructure of pollen can be either family, genera, or species diagnostic. The reason why we use light microscopy, scanning electron microscopy, and transmission electron microscopy to investigate pollen grains is that each type of microscope adds different important information that can be used to differentiate pollen. For example, here, using the LM, we are looking at more or less the outline and form of the pollen grain and the aperture type. Here you can see a spherical pollen compared to two more flattened types, a single aperture compared to 3 apertures compared to 6, etc., etc. Using the SEM, we are focusing on the surface of the pollen grains, trying to get detailed pictures showing sculpture types. For example, if the sculpture of the pollen is perforate, if it's reticulate, or echinate. For the TEM, we first need to embed the pollen grains and then make sections. These sections are then carefully studied, and we try to observe the thickness of the pollen wall, how it's structured, and its different layerings. What I have here in front of me are a few examples of potential sources for sampling. Water can be a source for pollen. Water, for example, from a puddle, from a lake, from a river, or even from the ocean. Even ice is a potential source for pollen. You can go on a glacier, take a drill core, take it home, and analyze it for pollen. When you, for example, are in the lab with water, it's easy to process. Just have to centrifuge it and then maybe do a fast astrolysis and you already have your pollen sample. Another fast way to get pollen is from honey. Honey usually has a lot of pollen inside, and this is also quite easy to study. You usually take 2 or 3 spoons of honey and you stir it in hot or boiling water. The honey will dissolve, then you just wait a bit and the pollen will settle at the bottom. Pour off the liquid. If you want, you can then astralize shortly the pollen and you can study it, so it goes quite fast. Here's also a typical sample. This is from a forest floor, but as soon as you have minerals in your sample, things get more difficult. You need different chemicals, etc. So if it's a sedimentary rock like this, or if it's a loose, recent samples from a forest, or sediments from a drill core, you are already now depending on chemicals. So for example, you need HCl to dissolve the carbonates, and you would also need HF to dissolve the silicates before you can isolate and extract your pollen or palynomorphs. Another interesting subject, of course, is here, for example, studying animals. There are many animals that are pollinators, so they have pollen on them. They are also easily worked in the lab. You can either just put it straightly under the microscope and pick out the pollen individually, or you can wash the insect with water, put them in a small tube, shake it a little bit. You can also use a brush to get the pollen off, and these can then be analyzed and studied relatively quickly. For recent material, of course, you just can go into the botanical gardens, or if you are a botanist and you know your plants, you can collect them yourself and identify them. Take samples from the anthers, process them using fast acetolysis. You can also go to herbariums. Herbariums usually have good identified plants with species, genus, families, etc. Many of the herbarium specimens also have then flowers where you can take samples and study the pollen.
Hello, my name is Christian Geier. I'm a PhD student here at the University of Vienna. So this microscope is special because there's a large distance between the objective and the microscope slide, which allows me to insert this micromanipulator between the object and the lens, and I can manipulate the pollen grains around. It's also a direct image microscope, which means if I move the table left, the image also moves left. So you can see if I insert my hair here, it will be seen also from the right side. Traditional microscopes invert the image and it's very difficult to manipulate it again. And now I show you how I pick them up. So I pick out the pollen grain with the nasal hair again from the glycerin drop. I have it now on the micromanipulator on the nasal hair, and I take the micromanipulator and the pipette with the ethanol, and then I can place a tiny drop of ethanol onto the stub and then the pollen grain into this ethanol drop.
What you see here is our spotter coater, and Christian will now show us how to put in the sample.
So the stub that we just made is here, and we place it in the machine. So after these 3 minutes or 4 minutes, the sample is covered in gold, and the aluminum is now covered with this thin layer of gold and is now ready for microscopy.
Hello, my name is Johannes Buchel. I'm a senior postdoc here at the department. My focus is on Northern Hemispheric palynophloras, and I use light microscopy and scanning electron microscopy to investigate pollen grains.
Could you just explain to us, just very simple, how this machine works?
So here we have the vacuum column, and here at the top is the cathode. The electrons are emitted and sent down here. They are then focused using electromagnetic lenses, and down here they hit the specimen. Secondary electrons are emitted, and these are then detected by the detector, and so we have here the image. And now we zoom in. This is now 4,000 times magnified. As you see, this has very nice sculpturing. And now we will take a picture. So now the electron beam is rastering the sample, and for taking a photo this takes a little bit longer. Now we zoom in and focus on this area here around the corpus. We are now at a magnification of 20,000. Now we focus and we take a picture. The electron beam now scans the specimen, and we get a high resolution picture of the surface sculpture.
Hi, my name is Carola Progina. I'm a master student at the University of Vienna, and in the course of my master thesis I'm investigating the polyneria of orchids with the help of light microscopy, scanning electron microscopy and transmission electron microscopy.
Carola, could you please explain to us just briefly how you process and handle your samples?
Yeah, to investigate the ultrastructure of my polynya of the orchids, first of all an embedding is necessary because otherwise we can't cut it with this ultramicrotome in order to see it in the transmission electron microscope. So first of all, the samples had to be dehydrated, had to be fixed and then embedded in a resin, which takes about 4 days. And afterwards, a resin block results, which the specimen inside. You can see the specimen here, this black sample. In order to be able to cut this resin block with the ultramicrotome, we first have to trim it. Therefore, we take a razor blade and trim the resin, which is too much. And in the end, we should get a trapezoidal form, which only contains the sample itself. After the block is trimmed, it is put here in the ultramicrotome to get cut with a thickness of about 70 to 90 nanometers. So the specimen holder moves against the edge of the knife, in this case a diamond knife, and afterwards they get fished with this loop. The sections are then placed on the grid under the binocular, and the water is removed with this filter paper.
Hello, I'm Silvia Ulrich and I'm working as a technical assistant and lecturer at the Department of Botany and Biodiversity Research.
This is our transmission electron microscope. There's an electron beam which is coming here through electromagnetic lenses and into the specimen block. So this is the section here, which is in this position, and the whole chamber is into the vacuum. So the picture is made from the electrons which are in interaction with the section, and then coming through a scintillator, and the final picture is then displayed here on a screen, but also here on our computer. Following the ultramicrotome, we have our semi-thin sections here in this box. And if you want to change the sample, we have to open our box and we are taking out one of our grids.
So Silvia, now the samples is firmly in the machine, and before we start photographing, what is the magnification that you're looking for? If you have a section of a pollen, what is the magnification that you're photographing?
So depending on the size of the pollen grain, usually the overview is done by about 3000.
Does that mean that you can make a picture showing a section through a whole pollen grain and also details then from part of the wall?
Details of the pollen grain we are usually making with a magnification about 12,000 or maybe also higher if needed, so 20 or 30,000, but much higher it's not necessary to use a higher magnification. So here we see a cross section through an enter of Ephedra. What you can see here is the overview over one single pollen grain inside the enter, and we will make a picture now of this pollen grain. This machine, it can also make high-resolution pictures at a magnification of 7,000. So we are making big panorama pictures, and we have a nice overview, and many different pollen grains are on this panorama picture.
As an archaeologist, your first question might be, why do I need pollen? What is it good for? Well, here are just a few suggestions. First, you could use pollen to investigate past vegetation and past climate. It could also provide information about a potential food source of your subject.
It could also give you some information about plants used, for example, in medicine, or it could tell you something about plants used in some sort of rituals.
And now we show you an example from an archaeological site.
This is an example of archaeological palynology, a case study from Pompeii. The project started in 2008 and it was the first analysis of pollen preserved in volcanic ash accumulated in the nasal cavities of human skeletal remains at Pompeii. The intention was to solve the problem of the Vesuvius eruption time in the year 79 Anno Domini by analyzing the pollen content. Samples of the volcanic ash for pollen analysis were chosen and collected by Marci and Renata Henneberg from various parts of the human skeletal remains stored at the Terme del Sano in Pompeii. Among possible places in Pompeii at which reliable samples of ash could be taken are the inner spaces of human and animal skeletons. These spaces, especially if protected by a complicated anatomy, often remain unspoiled by contamination from outside materials. The second reason for that choice of sampling is that the ash covering the body of the victim during the eruption and filling in the skeletal openings after the soft tissues decomposed has the closest composition to the ash at the time of eruption. Some of these bodies recovered during the first systematic excavation in Pompeii before being exposed to the outer environment were the first cast by filling spaces surrounding them with plaster of Paris, as you can see here. Some of these casts were discarded as unsuitable for public exhibitions, but the bodies inside the cast with the ash surrounding the bones are a good source for uncontaminated samples and can be used for palynological investigations. Nasal cavities prove to be a good source for collecting pollen, as you can see here, these inner spaces. Victims immediately prior to their death inhaled the air full of ash and other materials like pollen. Some of these would be retained in nasal cavities as demonstrated in forensic case studies. Even in skeletonized corpses, pollen can be retrieved from the nasal cavities. Assuming that the volcanic ash preserved in samples collected in the earlier mentioned way contains pollen accumulated in it during the eruption, we attempt pollen analysis in order to establish what species of plants are represented and in what quantity. This may provide us with a possible time in the year the eruption occurred. Previously, it has not been clear whether pollen grains remained intact in the pyroclastic flow that devastated the two Roman towns of Pompeii and Herculaneum. In previous studies, it was reported that pollen concentrations in soil samples from the AD 79 level in Pompeii were low and that pollen was poorly preserved. And only about 100 pollen types were found so far. Many of them has been reported to be crumpled. So we wondered if pollen mixed with the volcanic ash is destroyed or preserved intact, adhering to human skeletal remains. And also if fire has an effect of the pollen wall or grain, like pyroclastic flow, that may destroy pollen in the samples. So this is how the samples looked like. And for a first quality check of the samples for our investigations, we just washed the material, and as you can see here, we saw that the pollen grains in the samples, like here, was accumulated, but it remained intact and it was well preserved, so we could continue with our study. Here is an example of the pollen grains found in the child's nasal cavity after washing the isolated material. Besides primary minerals, which are typical for volcanic ash, like volcanic glass, the detected pollen grains were in a good condition and colored brown. As typical for subfossil and fossil samples, the brown color of the pollen walls may also be a consequence of the pyroclastic flow as heat stains the pollen wall brown. Here are some other examples of myrtaceae which we found, and also pine pollen, or as you can see here, artemisia. To be sure about the results, we made an experiment with pollen of Hedera, where we heated up pollen in water over a candle light to demonstrate the effect of heat of the pollen wall. Only a few seconds of heat makes the pollen wall already brown. Here, compared to the hydrated status in water, you can see a quite intensive brown color. After more heating, it's getting more intensive. So here you can see 30 minutes of heating up, and after 5 minutes of heating, the pollen grain is intensive brown, like our acetylized pollen grains. Here is an example of the pollen grains found in the child's nasal cavity after washing the isolated material. So the pollen grains are light brown, so they are not acetylized, but as you can see here, they are well preserved. So this is an example of Quercus pollen or OeAW pollen, typical for the region, but we also found Hedera pollen and Dipsacacea pollen, so the samples have been quite well preserved. So here you can see another example of pollen grains found in the adult's nasal cavity after washing the material. So again, pollen grain is light brown, it's not acetylized material, and the pollen grains are so far well preserved. So here's just some examples we found, like Cetrus pollen grains, some Pinaceae pollen, or Poaceae pollen grain, or Juglans pollen, so some wind-pollinated species. We also found Mimosaceae or Amarantaceae, or like typical for this region, Olea, so some Oleaceae pollen grains. Some Asteraceae we found as well, like Artemisia or Ambrosia, and we also found Castania pollen grains. So the good condition of the pollen grains in the sample from deep inside the nasal cavities indicates that the bones and skins of the nose protected it sufficiently from the heat and aided the pollen preservation. So after the first tests we made, we finally started with our standard chemical treatment for fossil pollen, and these preparation steps involves for example, the hydrochloric acid, which removes the carbonates, and hydrofluoric acid is necessary to remove the silicates, and the acetylysis treatment is also important, especially the acetylation, so the second step of the acetylysis treatment, because this stains the pollen walls brown. And we usually, for fossil samples with a lot of stones involved, we have to do all these steps to isolate the pollen grains and to make the sample quite clear. So our samples were boiled in concentrated hydrochloric acid for 10 minutes, followed by hydrofluoric acid for 5 minutes, and again in hydrochloric acid for about 5 minutes. And afterwards we just acetylized the samples for 5 minutes. A heavy liquid separation using zinc bromide was made to remove the mineral content, as you can see here in these layers here, from the sample. So all the darker layers are our organic fraction. What we have to do is we have to put in the heavy liquid here to our sample, and then centrifuge it for 5 minutes, and afterwards the mineral phase is situated at the bottom here in the test tube. So this is the heavy liquid, the zinc bromide, and what you can see here, this is the organic fraction. So these are the pollen grains mixed with other organic remnants like fungi spores and maybe some charcoal particles, and this is the distilled water here. So what we do is we just transfer this organic fraction together with the water, with a pipette, into a new test tube, and then we have to carefully wash it 3 times before we can further use it. Here you can see the acetylized sample before and after the heavy liquid separation. So this is before. You can see all the mineral particles in the sample mixed with the pollen grains and also some other remnants. And after the heavy liquid separation, most of these minerals have been removed, and you can see also the pollen grains much clearer for identification. After the heavy liquid separation, glycerin was added to the remaining organic material and transferred on glass slides for light microscopic investigation. So this is the final sample then. The liquid inside is glycerin and the pollen containing organic fraction is visible here. So these dark particles are the pollen grains. So usually we are screening now the sample on the glass slide from line to line, from one end to another. And we are counting 300 or up to 1000 pollen grains per sample. And this is our light microscope. And you can see here that we can also make a picture documentation on all the pollen types we found in the sample. In total, we identified 31 pollen types in the investigated samples, reflecting the vegetation as reported from ancient Pompeii. We found insect-pollinated taxa, like Hedera, and most of the pollen grains are wind-pollinated taxa, like the rest. Most of the rest you can see here. It is assumed that the material gained from inside the noses is a combination of pollen inhaled prior to death and volcanic ash. The amount of pollen found in the nasal cavities is usually very small and predominantly originated from wind-pollinated taxa. Also, insect-pollinated taxa can be found in nasal cavities, as we know from literature, due to inhaling pollen within dust, soil or mud, but in much smaller quantities. So this is also known from forensic case studies. So this table shows you the most frequent taxa found in the investigated samples. And here's also a table which shows you the amount of pollen found in the samples in percentage. So for the 2 children, the added and the control, which is the soil sample. And the taxa, the investigated taxa we found, are listed according to their pollination system. So the majority of the plant taxa found in the samples are flowering from spring to early summer. Artemisia, so here's this pollen grain, Ericaceae, Erica pollen is not displayed here, but Hedera pollen had a few taxa in the sample which are flowering later in the year. The wind-pollinated genus Artemisia is flowering from July to October to almost late November. But due to its long flowering period starting in July, Artemisia is not an indicator for a late volcanic eruption. The Ericaceae pollen, which is not displayed here, was found in all samples but in very low numbers. So this is listed here in this line. So less than 1%, and based on its flowering period in March to November, this insect-pollinated taxon is not relevant in this connection. The only taxon of interest in connection with the time of the eruption is the insect-pollinated plant Hedera. So again, Hedera pollen is shown here. Hedera pollen occurred in all samples, but in significantly high amounts in the children's nasal cavities. So here are the numbers listed for the Hedera pollen. And as you can see in the children's nose 1 and 2, the numbers are much higher than in the adult and the control sample. So with a flowering period from September to October, Hedera might indicate that the eruption in fact occurred in October-November. Compared to other studies, the amount of Hedera pollen in the soil sample is still noticeably high, but other investigations showed that Hedera pollen in soil samples covered by lapilli from Pompeii was missing, or if present, the amount was low, with a mean of only 0.4%. So this suggests that the pollen content in the pyroclastic flow must have been quite low. And due to the fact that our soil sample, so the control here, contains subfossil pollen mixed with modern pollen, the results cannot be used as an indicator for a late volcanic eruption. The most surprising is the unusual high concentration of Hedera in the samples. The discrepancy between the amount of Hedera pollen within the children's noses and the nose of the adult, and respectively in the soil sample, is a very strong evidence that some amount of Hedera pollen was already in the children's nasal cavity before inhaling ash-containing pollen. From ancient times, Hedera has been well known as a medical plant for its anti-inflammatory, antimicrobial properties, among other uses. And here you can also see that Hedera plant is still growing in Pompeii. Also, Pliny the Elder described several of its healing properties, including the ability that took away the discomfort of the nostrils. And Hippocrates and Dioscorides also recommended Hedera as a cure. Young leaves as well as flowers and fruits were reported to be used to produce medicine for different purposes. So experiments in the botanical garden of the University of Vienna, as you can see here, this Hedera plant growing in the garden, have shown that pollen can end up on the leaves just below the inflorescence. When young leaves were used to produce medicine in ancient times, it is therefore most likely that pollen also ended up in the medicine. And here you can see the flowers and also the loose pollen grains probably on the leaf. And even in modern phytomedicine, Hedera is mainly used as a medication for the treatment of bronchial diseases. So due to the fact that the amount of Hedera pollen in the soil sample was much lower, with only less than 2%, the high values of Hedera pollen in the children's noses highly suggest the use of Hedera as a medical plant in pediatrics. To conclude, based on the pollen spectra obtained from the analysis of the volcanic ash, it was not possible to define whether the Vesuvius eruption in AD 79 occurred in August or later in the year, in October or even November. But it was the first direct evidence of the treatment of respiratory conditions with an extract of Hedera as a medical plant nearly 2000 years ago. The sampling method developed in Pompeii, including the choice of the nasal cavity, was used for the first time for archaeological purposes. And the case study was finally published in 2020 in the journal Grana, which is open access. And if you want to know more about this study now, you can have a look in it.
We reached the end of our presentation. I hope that we have answered more questions than we have raised. But if you have any questions or any ideas or thoughts about taking samples, processing samples, or identifying pollen, please contact us. And our contact details you can see here.